Salut tout le monde! On va commencer. Merci d'être là aujourd'hui. Je vais introduire Uta en français. On parle un petit peu français. Are you going to take questions in French or English?
I can try, yeah.
Okay, she can
Je
try.
vais, je vais essayer. Alors, on va arriver, je suis sûre.
Merci d'être là. Voici Uta, qui est mon ancienne boss au Japon, puis ancienne collaboratrice à Cambridge. Uta a fait ses études en son master en biologie des plantes à Cologne en Allemagne. Elle est allemande. Elle a fait un PhD à l'ETH de Zurich. Elle s'est promenée un peu aux États-Unis, un peu partout, avant de démarrer son lab à Cambridge en 2012, où elle est depuis. Elle est maintenant directrice du Crop Science Center à Cambridge. Puis 2025, c'est une vraiment bonne année pour Uta, qui a été élue à la National Academy of Science et puis la Royal Society, qui vient tout juste d'arriver. Je suis vraiment contente que tu sois là pour nous visiter et pour nous présenter des données vraiment récentes, puis des super belles images, des vidéos qui sont des premières mondiales sur le mycorrhizal arbuscule. Puis voilà, take it away.
Alors, merci beaucoup, Alexandra. Pour moi, c'est la première fois au Canada. Et les derniers 3 jours avec Alexandra, c'était vraiment comme un rêve. Alors, on a fait le rafting et était dans le spa. Et alors, la fête d'été en ville, on a fait tout ça et c'était énormément fantastique. Alors, merci à toi et merci à tous pour être là. And that's the moment where I'm going to switch language now, talking or sharing with you some of our recent work on the vascular mycorrhizal symbiosis. So I have designed the talk such that the first half is a little bit about our attempts to get better grip on spatially resolving the symbiosis, which is the development is highly dynamic, so and very, very complex. So to actually get a deeper insight into the molecular processes that underpin the development and the functioning of the symbiosis on a spatial level. I'm going to share that. And then the second half will be a little bit more towards really the ambition to take discoveries that we make in the lab into, or at least bring it towards the field. So it's a lot. I have a lot put in, so I better get started. Just a quick reminder, I'm pretty sure being here at the cradle of one of the protocols that really revolutionized our field, the work by Pichet, Fortin and Bécard in the 80s, when they established the aseptic cultivation system. It really made all the difference, I think, to the field to be able to have controlled culture and controlled inoculum production, so that enables a completely different type of research. And it's a great honor to be here. And actually, I have already seen the protocol, the original protocol, which is quite impressive. So I'm pretty sure that there's very little introduction to arbuscular mycorrhizal symbiosis needed. So I put this picture up nevertheless. This is one of the most beautiful photographs ever taken, I think, of an arbuscule. So for those of you maybe a little bit less familiar with this kind of symbiosis, so the name, of course, comes from these kind of structures, which are the heart of the symbiosis, little trees, which ironically the fungus produces inside the plant cell. So here you see a little bit of the plant cell wall. So these structures are microscopically small. We're looking here at perhaps something like 40, 50 micron in length. So it's really tiny and absolutely spectacular. So sophisticated in structure. And ultimately, we're looking at in a form of compatibility and intimacy that really is quite extreme. Now, the outcome of the symbiosis, as the name says, is reciprocal benefit, which is manifested in the bidirectional nutrient exchange, the plant feeding the fungus with organic carbon, with photosynthase, and the fungus in return enhancing the mineral nutrition of the plant. So the fungus contributes to the uptake of water and minerals. And this is particularly pronounced for phosphate, And the underlying principle of the plant's benefit from the symbiosis is probably best illustrated here, where we're looking at a root living in a plant, so plant root living in an ongoing arbuscular mycorrhizal symbiosis. So all of these little threads here are fungal hyphae, the globally are fungal spores, and we can appreciate the extensive proliferation of the fungus in the substratum surrounding the root. So what we actually achieve in the symbiosis is a significant, a dramatic enhancement of the surface area for nutrient and or for mineral and water uptake. And as the fungus does that, it at the same time also proliferates extensively inside the roots, and then it is here at the arbuscles, as I mentioned before, where the fungus then delivers water and minerals to the plant. And when we focus on phosphates, this route is really extremely efficient. So this is a mix of data from some old work from the Smiths in Australia and ourselves in Rice. So what we're looking at here is the percentage contribution to the overall phosphate uptake by the symbiotic route. And what you find is that basically more than 70%, 80%, up to almost 100% across these really, really different angiosperms. So we have here representatives of 3 different dicot families and then the monocot rice. So across these diverse plant species, we find that the symbiotic uptake pathway dominates the direct route, which of course is also the plant can directly take up nutrients from the environment, but it is predominantly through the symbiotic route. And if we extrapolate this to what we know as well, namely that the symbiosis occurs in more than 80% of all land plants and in all terrestrial ecosystems, as this study shows from a little bit over 10 years ago, and combine now what we have learned through these radio tracer experiments that I showed on the last slide, we can safely say that the symbiotic phosphate uptake is the default strategy in nature, and it's a strategy that nature invented to secure plant nutrition. It was shown that the symbiosis actually does drive ecosystem productivity. So it's immensely important throughout the terrestrial ecosystems on planet Earth. So how about agroecosystems? So here we see actually the study that for the first time was able to put a number to the effect the symbiosis can have on yield in an agroecosystem. So this is work that came out of Rory Sawer's lab at Penn State. So he basically has 2 genotypes of maize that are isogenic. So they differ in one gene, which is mutated in the right-hand side. So this is a symbiosis-specific gene. When you mutate it and you grow the plants under sufficient nutrient supply, then the plant grows perfectly normal. So there's no pleiotropic effect. But when you grow now wild-type and mutant under this medium input subtropical field condition in Mexico, you can see with the naked eye that being able to engage in the symbiosis means that the plants are much better able to grow, there's better performance, and that also translates ultimately into a significant difference in yield. The ability to engage in the symbiosis gives you 30% more yield. So it is an important symbiosis not only in natural ecosystems. There is a potential to make better use of this of the fungi and the symbiosis for sustainable agriculture. So we cannot know enough about the symbiosis to just dive into some very beautiful biology, or at the same time also develop perhaps tactics that would allow to ultimately implement the symbiosis into sustainable agriculture settings for our future food production. So, but the problem or the limiting factor has really been the complexity with which this symbiosis presents itself. So here on the left-hand side, we see how the symbiosis develops when we follow a single infection site. So we have communication in the rhizosphere between the plant and the fungus. The fungus then eventually, when recognition and reprogramming for both organisms has occurred successfully, the fungus then penetrates into the root, grows into the inner cortex, never breaching the endodermis, never getting into the meristem, and then producing into individual cortex cells these arbuscles by evaginating the plant plasma membrane and by reiterative dichotomous branching, producing these spectacular arbuscles. And the production of the arbuscles, of course, is associated with monumental changes in the host cell architecture. So foremost to mention is the huge amount of membrane material that needs to be produced to envelope this growing structure. So the fungus and the plant cytoplasm remain separated by this symbiotic interface that is bordered by the periarbuscular membrane throughout the entire life of the arbuscle. And having said that, so despite what seems to be a really major investment for both organisms to accommodate or to produce these structures, they have a very short lifespan. So these are ephemeral structures that grow and collapse within the course of just a few days. But it is here across this periarbuscular membrane, across this symbiotic interface at the arbuscles, where the bidirectional nutrient exchange occurs, and when the fungus is well nourished, it then produces lipid storage structures, the vesicles, and it also grows out into the soil, forms this far-reaching mycelium, which is the site of nutrient uptake and also spore production. So we appreciate it's highly dynamic. And what this cartoon doesn't actually reflect is that it's also entirely non-synchronous. So typically we would normally observe that all of the structures, fungal structures, are actually present at the same time. So you are all experts. So you see here's a hyphopodium. We have the hyphae growing into the root. We have arbuscles, and then we also have vesicles. So if you wanted to, for example, study bidirectional nutrient exchange at the arbuscle, you're always dealing with the trouble of having all of the other structures present at the same time. So it's highly dynamic, highly asynchronous, and that somehow makes it difficult to really focus on molecular processes that would perhaps underpin specific stages of the interaction. So that led us to consider spatial transcriptomics as a means to dive into now tissue level type of, in this case, transcriptional activity of plant and fungus. And we work predominantly with rice in the lab. And we use the Resolve bioscience platform to eventually come in with a 100 gene set probe to now look at transcriptional activity of pre-selected genes. We chose some symbiosis markers, some cell type markers, and then genes that were eventually involved with the C, P, and N exchange. And what you see here is a section, a longitudinal section through a mycorrhizal root. We decided to go for longitudinal sections because we were hoping that we would catch alongside each other the different stages of arbuscle development and root colonization. And that is indeed the case and highlighted here. So on the left-hand side, you can see we have on the one hand growing arbuscle. Here's the growing arbuscle. We have the hypha in the middle, we have mature arbuscles, quite a number eventually, and then also the collapsing arbuscles and vesicles. So that work that we have, if you wish, almost like a time-resolved, definitely developmentally resolved set of stages. And you can also see here on the right-hand side the sort of nuclei of the fungi, of the fungus and the plant in neighboring cells. So all of these little dots, this is Duffy staining, are fungal nuclei and the plant nucleus here. So you can appreciate the huge size difference between plant and fungal nuclei. So, so now just a few data. So we included genes that we knew were encoding symbiotic nutrient transporters. So we have the symbiotic phosphate transporter, the symbiotic nitrate transporter, and the symbiotic fatty acid efflux transporter that feeds the fungus. So these are all from rice. And then at the same time, we have the fungal elongation factor 1 alpha, which is a sort of marker gene that is widely used as a housekeeping gene. And we were hoping with that to perhaps also outline the fungal structures, because the fungus is an aseptide fungus, so everything is basically one cell. So you would expect that the transcripts of a housekeeping gene would basically be lighting up the fungal structures in the root, and that is indeed the case. You can see here the hyphae eventually. You see vesicles, and then you see the fungal transcripts here in the arbusculated cell or in the arbuscle. And then, as was previously described in the literature by us and others, these are indeed now again patterns that have been described before, the transcripts of these transporters being specifically located and highly abundant in the arbusculated cell. So what we could show with that is that, which was very important for the company, actually Resolve was keen to see if we were able to detect plant and fungal transcripts in the same cell space, and that is definitely possible. Then we could confirm, so this was basically like a bit of a control, that these transporters that we were expecting to be in the arbusculated cell were there. But then the additional observation that we had not quite expected to be so dramatic is this total dominance of the phosphate transporter. I mean, if you look at the purple, the intensity of the purple signal relative to, for example, in this case, the nitrate transporter, there's a huge difference, which is actually, when you quantify it here, manifested in a whole order of magnitude more transcripts for the phosphate transporter relative to the nitrate transporter, or also the fatty acid transporter. And we know that phosphate, symbiotic phosphate uptake is really extremely efficient. So this transcript levels are basically going through the roof in the arbusculated cell. So also one slide to focus on the fungus. So we had 10 genes included for the fungus selected from the literature. 6 of these probes worked quite well, and we know near to nothing, or actually nothing, about spatial expression of fungal genes throughout that fungal syncytium. So we were super curious to see if there were any patterns that we could perhaps discern for the one or the other of this small number of genes. And I'm giving you here one example of HOG1, which is a fungal kinase. And what you can see, if you compare the pictures on the top where we have DAPI staining and we're looking at nuclei, and you look at the transcript here in purple, you see that actually there is a correlation between where these transcripts are labeled and the nuclear position. So there is a correlation between these kinase transcripts and nuclear position throughout the vesicles and throughout the syncytium. And that is not the same for other type of fungal genes. So what we can make of this is that there must be, as one could perhaps expect, different compartments within the fungal syncytium where different transcripts eventually would be differentially abundant. And that's hugely interesting because we know absolutely nothing about how the fungus is dealing with its transcriptional activity. Imagine you have a tube system which is continuous. The nuclei are zipping through. They're constantly moving. So how is transcription regulated? How is transcription actually occurring in the fungus? If you are in an arbuscle as a nucleus, or you are out in the extracellular, extra-radical mycelium, you need to express different genes, but you're also on the move all the time. So this is, it's quite fascinating to think about it. And the lab in Japan is actually now trying to get to grips with that. So here we have a huge discovery potential by focusing on the fungus producing a large list of, in this case, up to 500 probes can be used when we switch the spatial transcriptomic platform from Resolve to Merfish in this case. And so what we're doing there in collaboration with Alexandra is to collect fungal species, fungal transcripts, for that are available either in the literature or in our own data sets for asymbiosis and symbiosis stages. And in addition, we have more recently extended into single nuke RNA-seq in the fungus, in asymbiosis, or in colonized plant tissue. And then we collect all of these sequences together and designed our, it's about approximately 450, I think, probes that should hopefully allow us very, very soon to map out the transcriptional activity of the fungus. We start off with roots because we know how to embed the tissue, fix it, and so on for spatial transcriptomics. So we start from symbiosis, but we hope to actually be able to complete the picture asymbiosis and symbiosis. And Rhizophagus irregularis is our main fungal species. So that was formerly called Glomus intraradices. So that is the one that we can grow on plates according to the Laval protocol. So yeah, so I'm really, really excited about that. So that's a little bit of a outlook. So watch this space. There will be for sure something interesting coming out of that. So far, so good. One last slide to the spatial transcriptomic. I mean, there are lots of data that we have. This was just very recently published 2 months ago. And we made one observation, which I would like to share because it was something that we really didn't expect. And you can see what it is about already here without me saying much. You have this file here of morphologically equally appearing arbuscles. So they appear as if they are at exactly the same developmental stage, and they probably are indeed. And looking at the set of genes here for which we're monitoring the transcripts, these are all genes that have something to do with the bidirectional nutrient exchange. I mean, there's a transcription factor, then there are biosynthetic genes of the fatty acid pathway. There are transporters included. But what is really key is that we saw some of these arbusculated cells and arbuscles basically not showing any signal. So in the first place, we thought, oh, that's an artifact. I mean, you cut longitudinally and the root eventually grows like this. So maybe there's just no access to we cut such that we have no access to the cytosol of that set of cells. But that's not the case. We see that repeatedly. And what is the most compelling argument that these are totally normal arbuscles, they're just transcriptionally differently active, is that when we look at other type of genes, other probes, we see, okay, these arbuscles do, or the arbusculated cells do show signal. So this really, for the first time, taught us really that there is quite a bit of diversity if we look at arbuscle activity. And that needs to be factored in. If we normally do our classical kind of staining, which is invasive, and you just try to estimate what is the capacity, let's say, of your, for example, symbiotic nutrient uptake by getting an estimate of the percentage of arbuscle formation. I think we need to be a bit more careful in future because they are not entirely the same. So these data are consistent with non-uniform functioning of uniformly developed arbuscles. So that's quite interesting, and it also suggests at the same time that there's another level of regulation, because now if we see that we have actually neighboring arbusculated cells showing completely different transcriptional profiles, it means something is there that governs these changes. So that is interesting. So, and as Alexandra already mentioned in the introduction, so we were keen to add to this spatially resolved transcriptional activity, which still is static, right? I mean, we're killing the tissue, we're sectioning it, so we are able to look at different developmental stages of different arbuscles. But can we actually temporarily resolve colonization on the one hand, but then really importantly, arbuscle development and collapse. So is it possible to develop a cell biological approach that would allow us to follow the arbuscles from birth to death? And Jen McGalley is a postdoc in the lab who did a PhD also in the lab, and during her PhD, she developed this device. It was actually very sweet. She came back from Christmas holidays and she said, "Oh, Uta, my brother and I, we sat in the basement and we came up with this idea. We 3D printed this device and they seem to work for live imaging, non-invasive live imaging." And how does the system work is best shown here. So you have a small compartment. It's probably 1 inch by 1 inch wide. And you plant the plant, in our case, this is rice, inoculate upon planting. And there is a foot here that extends from that little compartment. So the roots would see the fungus here, would continue to grow into this foot. You can see roots here, and you can image this because this is basically the format of a microscope slide. So that now allows to do non-invasive imaging. So Jen published this 2 years ago, and the code for the 3D printing, for anybody who's interested, is in that paper. So it is quite widely used already. So it's making its way. So in the first place, let me quickly tell you what are the type of fluorescently labeled proteins that Jen is using. So we have stably transformed rice lines that express an RFP-tagged symbiotic phosphate transporter, then a split venous combination of the 2 fatty acid efflux transporters. These are half-size ABCG transporters. And then we have a membrane marker, a periavascular membrane marker, a SCUMP, which just highlights the whole membrane from the beginning of the arbuscle formation till the end. So Jen, in the first place, just tried the system. So here we're now having a line where we combined, so it's in the same line, you have a double reporter. So you have the membrane marker and a symbiotic phosphate transporter. And now you can already see a pattern emerging. I mean, this was something that we expected because, again, we're in the first place confirming what was published by different methods in the literature. So you see the membrane marker around the invading trunk, and it continues all the way to the collapse of the arbuscle. You have the stages outlined here by the cartoon on the top. And you see that there's no phosphate transporter here. It starts to come up as we start to see fine branches. So here we have a cross-section. This is an intensity plot. That is the cross-section through the trunk. And you see only the membrane marker, but not the PT11. It's not there. Whoops, yeah. And then when you do the intensity plot through the fine branches, now you see PT11 coming up. And that continues as the arbuscle produces more fine branches. So we confirm that PT11 localizes to the periarbuscular membrane surrounding arbuscular fine branches. And then as the arbuscle collapses, PT11 is very quickly removed from the periarbuscular membrane. But then Jen also made this fascinating observation, which is that she looked at 2 neighboring arbuscles here, which are developmentally, again, at an equivalent stage. And when you look at the intensity of the PT11 RFP signal, you see there's a lot of PT11 here, but not in that top arbuscle. And when she measured the ratio of RFP to GFP, then you could see, I mean, you have very different abundance of PT11 in these 2 neighboring equally developed arbuscles. And that is, of course, reminiscent of what I just told you about the spatial transcriptomics. So we collectively observe on the transcript and protein level that there's something that we like to call arbuscle individuality. So there are differences between individual arbuscles transcriptionally and on the protein level. Okay. So now having showed you that, these are all still individual snapshots of different arbuscles. So can we now actually go into a approach where we follow the arbuscular development or the arbuscular life from birth to death. And here it is. So Jen did a marathon of 56 hours where she every 2 hours imaged the same tissue section, the same cells. And as you can see here, so there is a set of cells that all became eventually arbusculated. And over this time, you can see how beautiful these arbuscles develop, and also how quickly they eventually collapse. If you look at this one here. So it actually disappears within very, very short time. So this allows us now to ask a whole set of new questions. So one was, of course, what is actually the distribution of lifespan across individual arbuscles? So some turn over very quickly within 24 hours, and others would perhaps span up to 4 days. But what is interesting, there's never a static stage. So even those that take 4 days, they would continue to grow and then at one point just initiate collapse. But they don't sit there. It's not that the arbuscles form and then pump, and then collapse. It is really like a breathing. It's formation and collapse, all the time in move. So Jen then also was interested in now mapping protein abundances against that spatial temporal dynamic that we now can follow. And she, of course, was interested in the cost-benefit nutrient transporter. So these are the phosphate transporter and the carbon efflux transporters. And here we're first looking just at the membrane marker. So this is our arbuscle, a fast one, turns over within 24 hours. So now let's map onto this, the symbiotic phosphate transporter. Here we go. Tack, there it comes. When the fine branches come, the phosphate transporter comes. So how about mapping on top the fatty acid efflux transporters? Well, here we see a different pattern. Now it's the phosphate transporter, but the efflux transporter is there from the beginning. They are already on the perihyphal membrane surrounding the trunk of the arbuscle. And so you can, of course, now break this down into different stages of that arbuscular development and really map the expression of proteins onto that dynamic range. And it's quite surprising to see that there is not a strict co-regulation. We would have expected that carbon efflux and phosphate uptake are linked, and they're not strictly linked, which is interesting. So we hadn't expected that. The other really important aspect I find about this work and this data set, because the arbuscle is never static, and these transporters are both, or all three, immediately removed from the periarbuscular membrane when the collapse initiates. So that means that the transport, the exchange of the key nutrients that underpin the mutualism of that symbiosis is entirely coupled to the dynamic process of hyphal growth and branching. So it's coupled to the active elongation and branching, which is, I think, quite interesting as well, because we always ask ourselves, why are the arbuscles collapsing? So if you accept that you need expansion, once the arbuscle fills the cell, it's done its job. So it's probably removed by the cell. So that's what I told you. So far, symbiotic nutrient exchange is linked to dynamic arbuscle growth and locally regulated. We take a deep breath and we turn a page. So for the last 10 minutes, I would now like to move a little bit towards this, well, at least an outlook to translation opportunities. So having told you that there is an obvious positive effect the symbiosis can have on crop nutrition with that picture of the maize field, you may wonder why we haven't made better use of the symbiosis already as a biofertilizer, as a replacement for fertilizer application. And the reason for that, one of the reasons for that is shown here, which is from an extremely old publication, late 60s. And that is that there is an inverse correlation between the amount of phosphate or generally fertilizer or mineral nutrient availability in the surrounding of the plant and the extent of colonization. So in other words, the better the plant is nourished, the less eager it is to engage in the colonization. And so that under agricultural settings is really fiddly to work with. So it's not an easy trait, mycorrhizal responsiveness, to make use of, because it's relatively strongly environmentally regulated. So I think we may have developed an entry into a strategy that could help us to better implement the symbiosis by basically making colonization more robust. So that will quickly walk you through that. So that is relying on some years of signaling work. So the approach that I'm proposing in the end is engineering. So we discovered a receptor mutant quite a number of years ago that is entirely unable to engage with the fungus, as you can see here. So you can detect the fungus in the vicinity, but the fungus does basically not engage with this route of the symbiosis at all. So we first had the mutant and then cloned the gene, and it turned out to be this very uninspiring dwarf 14-like gene, which was described in the literature, so we had to stick to that name. But it actually corresponds to the Arabidopsis gene, which has a different name, which is called carrageenan sensitive 2. So it is the receptor for carrageen. Carrageen is a smog constituent. So it is the receptor that is required in fire-following plants to initiate seed germination after bushfire. And so here we are. This is our receptor for symbiosis. So that was a bit surprising. What is interesting about this receptor is also the evolutionarily older paralog of the strigolactone receptor. So early diverging plants had only this receptor, and then the strigolactone receptor evolved from it later. So what was known from the literature, predominantly Arabidopsis, is that this receptor complex looks as follows: that when the receptor is activated by ligand binding, it recruits these other two proteins, an F-box protein, which in rice is called D3, in Arabidopsis is MAX2, and a negative regulator, SMAX1. So upon activation of the receptor, this complex forms. The F-box protein leads to the ubiquitination of the negative regulator, which is sent for degradation to the 26S proteasome machinery, and thereby de-repressing developmental programs such as seed germination in fire-following plants. So as you can see, looking at the involvement of these different components in symbiosis, we basically reproduce the requirement of these, or the function of these different components also in symbiosis. If you have the receptor or F-box protein mutants, you lose symbiosis. If you remove the negative regulator from the system, you get hypercolonization. So that indicated to us that perhaps the strong phenotype of the receptor or the F-box protein is due to stabilization of SMAX1, right? If you don't have these two functions here, the negative regulator would be stabilized. And that might underpin the phenotypes that we see here. And indeed, looking at a Western blot, that is exactly what happens. In the mutant, you have a stabilization of SMAX1, which is not occurring in the wild type. So all of that is long time published. But John Min, who was a postdoc back then, she was then interested to figure out what is it that SMAX1 is blocking here. And so she did a transcriptome analysis across a number of different genotypes and just extracted here what is important for us to understand SMAX function. So she found an overrepresentation of genes that are biosynthetic strigolactone genes. And we then collaborated with a lab in the Netherlands to also measure strigolactone and to just focus on the important bit of that graph. Here we have the D3 mutant, so this is the F-box mutant. This is the amount of strigolactone in the F-box mutant, which is similar to the wild type. If you bring the SMAX1 mutant into the F-box mutant background, you see now that you have the significant increase in strigolactone. So what we see transcriptionally changing indeed is also physiologically translated. The other overrepresentation she found was of genes that make up the common symbiosis signaling pathway. So common sim means this pathway is required in legumes for the engagement with nitrogen-fixing symbiotic bacteria, rhizobia, and for mycorrhizal interaction. So that means each of these common symbiosis signaling pathway components, when you knock them out in legumes, you lose both endosymbiosis. So, but all plants that do mycorrhizal symbiosis have this pathway as well. And here in rice, when you knock out SMAX1, you get a massive overexpression of these genes. And we then wanted to verify that, that indeed this pathway is upstream of the common symbiosis signaling pathway. So we introduced a gain of function, so a dominant allele of a central kinase in this common symbiosis signaling pathway into the receptor mutant, and were able to restore colonization. So that confirmed that the agristatic relationship is such that the D14-like signaling pathway SMAX1 is upstream of common syn. So that brings me to the model that we then had developed. So basically, when the receptor is activated and the receptor complex is recruited, the negative regulator is degraded and must liberate transcription factors that we don't know that now drive strigolactone biosynthesis. So strigolactone is released into the root and into the rhizosphere, and actually activates the fungus for symbiosis and navigates it to the root. So we have the calling out mechanism for the fungus activated, and at the same time we open the door for accommodation of the fungus because we activate the common symbiosis signaling pathway. And accordingly, when this is happening in a normal way, you have normal colonization. If you genetically remove SMAX1, you get hypercolonization. In the unfavorable scenario, D14-like is not activated. There's no degradation of SMAX1. It keeps blocking these elusive transcription factors. We don't have strigolactone biosynthesis, and we don't get the symbiosis program activated, and accordingly, we lose symbiosis interaction. And the question now, of course, is this what is happening under high phosphate? And Thomas is a postdoc in the lab who designed a Degron sensor to monitor the stabilization of SMAX1 in planta. And this is basically a relatively simple approach. You overexpress the critical domain of the SMAX1 protein, critical for interaction with the receptor, translationally fused to a fluorescent protein. And then you have another fluorescent protein, but these two proteins are separated by a viral motif that makes the ribosome drop off and come on again. So you have one RNA, but you get two proteins at equal ratio. So you can look at stabilization or degradation of this protein relative to that. And here we have a variant which is constitutively stabilized because the deletion of that motif renders it unable to engage with the receptor. And this all worked very beautifully. Here we're looking at the protoplast data. So you can see the YFP is there, but actually the RFP is not there. So basically SMAX1 wild type domain is degraded. When you stabilize, when you use the stabilized version, you have stabilization in the nucleus, both have an NLS. And so that allows us to now monitor stability of the SMAX1 protein. And so Thomas, of course, produce stably transformed plants. I don't want to dwell too much on this. So he's characterized the pattern of SMAX1 stability across the rice plant. And what is perhaps most interesting for us in the terms of symbiosis to consider is that when you have a lateral root emerging, so the cooler the color, the less stable the protein is. So you see you have a higher degree of stabilization. So every dot is a nucleus expressing the protein. You have a higher stabilization here and a lower stabilization there, and it is the lateral roots that are predominantly colonized. So this is quite interesting. And now the key experiment, if we grow these Degon lines under two different phosphate regimes, so either 25 micromolar or 250 micromolar, there's a number of things that we observe. So on the one hand, and this was the key thing we wanted to know, if we take the wild type SMAX1 and or determine the stability ratio at low phosphate and at high phosphate, we see that there is a significant increase in stability. So yes, indeed, phosphate regulates SMAX1 stability such that higher phosphate leads to an increased SMAX1 stability. But there's the other thing that we observe is also in the first place as expected when we have the mutant version, the stabilized version, we see how much more stable that mutant version is relative to the wild type version. But also the mutant is actually still regulated. The stability is still regulated in response to phosphate. So there's another pathway for sure. And that now allows us, knowing that there is a link to phosphate, to actually introduce SMAX1 mutants into a set of different rice varieties that are for the one or the other reason interesting in a sort of agricultural context. And we indeed can, across a number of different, actually any rice cultivar that we have used so far, whether Japonica or Indica, we can always stabilize colonization under non-favorable conditions. So this is a good step, a great step forward, and SMAX1 is our engineering target. We're having a whole project on that with the plants currently in the field in Colombia, because these are gene-edited plants, and at the time that was a good destination to try their field performance. So that brings me, to the end. So indeed, Uta, indeed, we have under low phosphate condition, the whole pathway active, and under high phosphate condition, a sort of reduction of engagement with the fungus, which is at least in part mediated through D14, like definitely mediated through SMAX1 with an unknown component still to be discovered. But I just would like to finish off the presentation by saying or emphasizing what an elegant way of linking the phosphate status of the plant with, the integration really of the phosphate status of the plant with the production of strigolactones to core for the fungus and opening the door basically for the fungus to accommodate. So all integrated through one pathway. And well, what I basically told you today is that there are multiple levels of control of mycorrhizal development systemically and on the tissue level, and eventually even cell autonomously. And that brings me to the end of the relatively long talk. So I just wrap it up really quickly. So this is the Cambridge gang, and here we see the different type of research lines that we have in the lab. Here is the Japanese group. Hector has left in the meantime, and I'm super grateful to a number of fantastic collaborators, former members of the lab, and we've been well funded in recent years. Thank you.